Okay, so we'll make a start because we're fairly time tight. This is one of the sessions where people who have been funded by donations from you and the CJD Foundation to do their research, and the structure is the before people, each of whom will talk for 15 minutes, and they have been instructed, I hope, to talk to you, the families, to explain what is technical in a way that you should understand. There'll be no question and answer session in this particular room. This is postponed to a later thing when we're back in the main ballroom. So questions, if you want to ask any questions, certainly you could write them down to remind yourself, and we'll deal with them later. So without further ado, I'll move on to our first presenter, Thomas Cunningham, from the MRC Prion unit. And you can see his topic here. I've been asked to give a kind of very brief, general sort of description of where this kind of research fits in. And I think, obviously, as you're all probably aware now, genetic prion disease is specifically caused by a mutation in the prion gene. But the commonest form of CJD is sporadic CJD, which is so named because it occurs sporadically and is not due to any specific gene mutation. And its cause remains essentially undetermined. But genetic variations in the person do affect the risk of your developing sporadic CJD. You'll have all heard by now at least of the codon 129 variation, where if you are so-called MM in that variation, you are at more risk of developing sporadic CJD, and if you're MV, you're at less risk. Obviously, one of the questions about sporadic CJD is, are there other genetic factors which are variations in the population which might influence the risk of your getting disease? And you heard a bit about this from Simon Mead. And this is about one particular gene, STX6, which is certainly a candidate for affecting risk for sporadic CJD. So, without further ado.
Right, thanks very much. And first of all, I'd really like to thank CJD Foundation, the organisers and all the families, and for all the great fundraising and for supporting this work. So I lead the mouse models of prion diseases program at the MRC Prion unit in London at UCL. And the study I'm going to talk about today is a joint effort between my lab and Professor Simon Mead's lab. And in particular, it's the work of a really talented PhD student, Lizzie Hill. And my title today refers to generating new STX6 overexpression mouse models, and I'll get to that a little bit later in my talk. But I'm also going to give you some of the wider context into where that fits in with our STX6 studies more broadly. So first of all, you heard a little bit about Simon, but I'll refresh some of that. So syntaxin 6, or STX6, what is this gene? It's a protein that regulates transport or trafficking of proteins and other molecules around the inside of a cell. So this diagram serves to illustrate it's actually very complex. There's a lot of complex transport going on inside cells, and that transport could be recycling of proteins or for degradation, for example, such as the prion protein or prions, and it could be delivery of cargo to various parts of the cell for normal physiological functions. And STX6 is one of many proteins that form part of this transport machinery. And so why are we interested in this gene? You've already heard from Simon that human genetic studies discovered that one particular version of the STX6 gene present in the human population increases risk for developing sporadic CJD. And so this diagram, Simon's already showed it, but this is a Manhattan plot to show that genetics data, and we can see this STX6 version, I'll call it version A, sort of rises above those variants that do not have risk. And if you have this particular version of STX6, you actually have higher levels of STX6 in the brain. And that together confers a small but statistically significant risk for developing sporadic CJD. And as Simon said, the risk is still quite low, even if you have this variant, but this strong association tells us that this gene is involved in prion disease biology, and we really want to understand how. So one way of studying that is using animal models, and one of the best models we have is mice. And so why do we use mice, you might be wondering? Well, actually, 98% of genes that are present in humans are also present in mice. And most of those genes perform the exact same function. So mice have a prion protein gene, and mice are susceptible to prion disease. Mice have an STX6 gene, and that gene does the same function in transporting molecules and proteins around cells. The second point is that our mouse models are really excellent. They recapitulate all the hallmarks of the human disease in a relatively short time frame. So that includes buildup of prions in the brain, development or upregulation of relevant biomarkers, neuroinflammation, and neurological decline. And these measures, these signs are really important, and they're really needed for when we're studying factors that we think could modulate the disease. So that's the importance of animal models. And then the final point is we can now actually relatively easily genetically modify mice to model human genetics findings, and that's exactly what we're doing with STX6. And so the first approach for a mouse geneticist such as myself is, when we're understanding gene function, is to what we call knock out the gene. So that's to physically cut it out of the animal's DNA. And this slide just shows you how we do that. So first, we harvest fertilized eggs, so single-cell embryos from early-stage pregnant mice, and we can micro-inject them with something called a CRISPR-Cas9 complex. And this complex is essentially a pair of molecular scissors that we can target to any DNA sequence that we want. So here we've targeted to the STX6 gene to essentially cut it out of the genome. And then we reimplant those embryos back into mice, and then we screen the offspring and try to find those pups that have the deletion that we want, and we can then breed a colony of mice to be able to study what removal of STX6 does. And so the question we're asking is, in our prion disease models, what is the impact of having no STX6 on the etiology of the disease in the mouse? And from our first experiment, actually the answer was removal of STX6 had no real meaningful impact infected with our usual 1% prion dose. So these mice developed prion disease within a similar time frame to controls. They had similar levels of infectivity in the brain throughout disease. They had similar pathological changes in the brain. So then we had to ask ourselves, what could we be missing? What's the missing link? And actually our hunch was it could be related to the dose. So a 1%, as we call it, 1% prion preparation is actually a very high dose of prions. And the reason we use that very high dose is because it's really reliable, really robust. 100% of mice get the disease in a very consistent manner. And excuse me, and oops, sorry, lost my way there. And so that enables us to study late diseases very well. Sorry. There we go. That enables us to study very late stages of disease very well. So rapid replication and spread of prions through the brain, neurotoxicity, and neurological decline. But it actually bypasses some of these early disease stages. So we can't measure, for example, how prions establish early pathology and how they begin the process of escaping those first infected cells in the brain. And so to better model those very early stages, we set up what we call a serial dilution experiment. So this is where instead of injecting the usual high 1% dose, we can inject a series of dilutions, and we actually dilute up to 1 million times that 1% dose. And then we measure what we call the attack rate. So what proportion of mice become infected as we dilute down. And so at the lowest doses, the attack rate, I should say that 1% that has a 100% attack rate. And then when we get down to those lowest doses, the attack rate gradually decreases, and that's what we would expect to see. Meaning that some mice are able to clear out those low doses of prions before they take a hold in the brain. Whereas in our STX6 knockout mice, this is where we see a protective effect. So this is the blue bars on the chart. And so there we see a statistically significantly, I can't say that properly, but it's, we see a significantly lower attack rate in the STX6 knockout mice. So to put it another way, removing STX6 reduces the chances of a low dose of prions from taking hold in the brain in these mice. So we think really that STX6 has a role to play in very early disease stages, in those initiation stages. We can also change STX6 levels in cells in a dish and measure the effect on prion infection. And so as we do in animals, we can genetically lower STX6 levels and ask at the cellular level in a bit more fine detail as to what the impact of this gene is. So in control prion-infected cells, we see the prion protein is distributed around the outside of the cell at the cell surface, and that's what we expect to see. Whereas when we reduce STX6, we see this interesting change in distribution of the prion protein towards the interior of the cell. And so that seems to us to be consistent with the role in cellular transport, and that's something we're thinking about trying to follow up to understand that better. And so now going back to mice, and now what we're doing is we're making models where we increase STX6 rather than remove it. And to do this, it's a similar process as before. We take these single cell embryos, and this time we inject it with extra copies of STX6, so little pieces of DNA that we've engineered in the lab. And when we inject those into the embryos, they insert themselves into the DNA of the embryos. So we have extra copies, and that increases the levels. And so where we are now, we've screened a large number of offspring from these injections, and we've now started to establish colonies with different levels of STX6. So we have our control mice, which have the usual 2 copies, and we have mice that have 15 copies, mice that have 5 copies, mice that have 3 copies. So what we're going to start quite soon is to put these mice through our prion transmission experiments and ask the question, what does increasing STX6 do to prion disease? And that mimics a bit more closely the situation with the human risk variant that increases levels. And onto my summary slide. So a natural genetic variant in STX6 increases levels of the gene and the protein and increases risk for developing sporadic CJD. If we remove STX6, this has a modest protective effect, but in particular, when mice are infected with a very low dose of prions, indicating the effect may lie with how brain cells deal with very early infection. Cell culture studies show a change in prion protein distribution, which is consistent with a role in cell transport. STX6 lowering, in a therapeutic sense, I don't think it looks like a promising candidate for treating established disease, but I think it's still really important to try to understand the mechanism of the protective effect potentially in those early disease stages, which I think it requires further investigation. And to that end, we've engineered new mouse models now for studying how increased STX6 levels affects prion disease to mirror the effect of that risk variant that also increases the levels, and to complement our mouse studies where we've investigated removal. And just so to thank the folks involved, so like I said, Lizzie Hill, a really great, talented PhD student. This is a collaboration between my lab and Simon Mead's lab, and then the Peter Clone lab, who provided support on the cell biology side, and also important contributions from our animal facility and histology facility. And again, a really big thanks to the families and funders and the CJD Foundation and the Medical Research Council. Thank you.
Thank you very much, and even ahead of time. That's good. As I say, if you have got questions, try to remind yourself of them, write them down, because it'll be in the question and answer session later on. Thank you very much. So the next presentation is from Victoria Lawson from the University of Melbourne, Australia. And again, I was asked to put a kind of frame around it. And as you'll all understand, I hope by now, if you didn't before, the core molecular abnormality in prion disease is the conversion of the normal prion protein, which we're all busy making just now, to an abnormal disease-related form. And without the normal prion protein, you don't get prion disease. I mean, if our prion protein was removed, we would be resistant to having the prion diseases. And of course, that's led to certain therapeutic approaches whereby if you somehow get rid of or reduce the level of normal prion protein, maybe that would reduce the production of abnormal prion protein, which could perhaps be cleared by cells, and therefore actually slow down or delay the disease. And there are a number of approaches which you have or will hear about that relate to this. But of course, we're all making this normal prion protein, so the question has always been, well, it's there for a reason, maybe we need it, is it harmful to remove it? Although having said that, sometimes proteins are important in certain parts of life and they're not necessarily important throughout life. But this the topic you're about to hear is one exploration of what PrPC might be doing in relation, for example, to cancer.
Great, thanks, Richard. Do my slides come up? Because I can see it here, but not there. Nope.
You have to advance it or should.
Oh, it's not in, sorry. No, yep.
Get there.
There we go. Rocket scientist. So it's a great pleasure to be here with you today to present an update of the work that we've been doing through your generous support. So as Richard just said, I'm going to update you on the risk of cancer from reduced expression of PrPC and the implications for prion disease treatment. So we're based at the University of Melbourne in Australia, so I'd just like to start by acknowledging the traditional owners of the land on which the work was performed, which is the Wurundjeri people of the Kulin Nation, and pay respects to their elders past and present. So my research group is we look at transmissible neurodegeneration, so most of our research is looking at prion and prion-like diseases. But the work I'm going to tell you about today is, again, focused on cancer. So this is my research group, and the cancer work that I'm going to tell you about has been performed predominantly by Ms. Shana Patelli, who is in the third year or final year of her PhD, and also some work begun by now Dr. Natalia Fortunato. We've also been really lucky to have some undergraduate students who have contributed to this work, so I just want to acknowledge them, and my collaborators, the Mantomatiotis group and the Holland group in Melbourne. So, again, to set the scene, I am talking about the normal cellular form of the prion protein, which misfolds in prion diseases, but I'm going to be talking to you about it in the context of cancer. And this is my summary slide, but I might come back to it because it's probably a little complicated to start off with. So, as you all know by now, that the normal cellular form of the prion protein, which I'm showing here, misfolds and leads to neurodegeneration, so prion disease. So in this context, what we're seeing is the normal cellular form of the protein misfolds and we get cell death and neurodegeneration. The flip side of this is cancer, where we have, instead of cells dying, cells proliferating and forming things like tumours in this case in a brain, but in other organs of the body. And the cellular prion protein can contribute to both of these processes. So really the question we're asking is if the cellular prion protein can contribute to both neurodegeneration and cancer, which are kind of two sides of the same coin, either cell proliferation or cell death, what is the impact going to be of therapeutics that might modulate the expression of the cellular form of the prion protein? So what we know from the literature already, the role of the cellular form of the prion protein in cancer is that it has increased expression in a number of different organs. So we can see increased prion protein expression in brain, breast, colorectal, gastric, pancreatic and skin cancer. And an example is shown here. Here's a control brain of an individual who doesn't have brain cancer, and here is the increased expression of PrPC in an individual with grade 4, so a very high grade glioblastoma. And the kind of connections that people have made is that when we see this increased PrPC expression, the tumour grade is worse, so a more aggressive cancer. It seems to be associated with tumour recurrence, reduced patient survival and increased therapy resistance. And so, again, talking about the role of PrPC in cancer, this is, if you're a cancer biologist, you would be so over this diagram, but basically this indicates the features or the characteristics of cells which can cause cancer. And a couple of the features that we see here, such as sustained proliferative signalling, enabling replicative immortality, resisting cell death, and becoming more invasive or metastatic or spreading, are also characteristics that have been linked to the normal function of the cellular prion protein. So it kind of relates that these things could be interconnected, and that PrPC has been associated with a number of signalling pathways, and those signalling pathways are also dysregulated in cancer. So that's the foundation of what we're doing. And the foundation of the work we were doing was really, again, to look at the two sides of that coin. So if PrPC misfolding causes neurodegeneration, but also can cause cancer, maybe if we understand how it causes cancer, that will inform the normal cellular function of the protein and will help with neurodegeneration. So now what I'm going to try and simply walk you through is how cancer works. So my example of cancer is going to be like driving a car. So if you think about driving a car, I want you to think about the brake and the accelerator. So hopefully for all of us right now, our brake is on and our accelerator is off and our car is stationary. So we are not developing cancer. A benign cancer or a low-grade cancer can occur when we have the brake off, but the brake is off and the accelerator is off. And if you're on a flat road, hopefully when you have the brake off but you don't put the accelerator on, the car remains stationary. However, hopefully I've got this right, if you take the brake off and you put the accelerator on, the car will drive forward, and that is like cancer progression, and that would be brake off, accelerator on. Okay, so that's basically, so I'm going to keep trying to refer to this as brake on, accelerator off is normal individuals, no cancer risk. Brake off, accelerator off, somebody who might be at risk of developing a cancer, but it's a low-grade cancer. And then when I talk about brake off, accelerator on, I'm talking about a high-grade cancer. All right, so when we talk about the brake and the accelerator, for the scientists in the audience, and maybe people have heard of these terms, the brake is a tumour suppressor gene. So typically what happens in cancer is that you have these genes that are called tumour suppressor genes, and they get turned off in cancer. So a little bit like Thomas was just talking about, chopping out a gene. In this case, this gene doesn't work and the brake is off. And then the accelerator is an oncogene. So the oncogene is like the accelerator. It drives the cancer forward. All right, so this is a flat road. What happens if we tilt the road? So what if we're on a hill? If we're on a hill and we have the brake on and the accelerator off, if your car is a good car, it's not going to go down the hill. However, if we have the brake off and the accelerator off and we're on a hill, the chances are that our car will roll down the hill. So when we introduce this stress, the car is now going down the hill. And then last of all, we have our brake off, accelerator on, and our car is already at the bottom of the hill, but it might contribute to it. So what we're looking at is whether or not the prion protein, the normal cellular form of the prion protein, is the thing that pushes this stress, that gives you this hill, that changes the risk of cancer when you've got just the brake off and the accelerator off and drives towards cancer. So that's the question that we were asking. So the way we're answering... Okay. The way we're answering this question is, I'm going to have to show you a complex version, is we're using a genetic model of cancer, and in this genetic model, our tumour suppressor gene is called P10, and our oncogene is called PI3 kinase. And so in this model here, this is a low-grade cancer. It's a tumour suppressor gene knocked out. It is the brake off. So we have a brake off model, and we have a brake off accelerator on model. So basically had, and the other benefit of this model, probably more relevant to the scientists in the audience, is that these are floxed genes, and so we can turn the cancer on when we want to. So it's not a case of the mice are born and develop cancer over the period of their lives. The mice are born, they get to an age where we're interested in manipulating them, and then we turn the genes on. That also means that we also have the normal version, which is the brake on and the accelerator off version. So first of all, we looked at an in vitro study. So what we can do here is we can take the brains of these mice, we cut out what's called the subventricular zone. It's a place where there are these regenerative stem cells in the brain. And what we can do is we can grow them in a tissue culture dish. And when we grow them in the tissue culture dish and we turn these genes on, we can look at what that effect is. So very simply shown here, when I knock out the prion protein, so in PrP null mice, what you see is if you have no risk of cancer and you knock out PrP, the size of the cells doesn't change, so it doesn't affect the car stays on the road. In the case of our at risk of cancer but low-grade glioma, what we see when we knock out the prion protein gene is we get a massive increase in the size of the tumour cells compared to wild-type cells. But really surprisingly, the opposite is true. So in this high-grade model where we've got the brake off and the accelerator on, it's the opposite. So when you knock out PrP, you reduce the progression of the disease. So it's complex. So this is just showing this in a little bit more detail. So again, same data. In this case, what we've got is the parental cell lines. So these are like having the brake on and the accelerator off. Nothing happens when you change prion protein expression. So if you're at risk of cancer, if you've got no risk of cancer, nothing's going on. But in the case of this low-grade glioma, when we knock out PrP, we get an increased size of the neural spheres and we get this increased replication of the cells. And again, in the high-grade model, this is the brake-off accelerator on model. What I showed before, it remains true. So when you knock out the prion protein, you get decreased proliferation, but you get increased, and this is the proliferation of the cells when PrP is expressed. So we've looked at all of these hallmarks of cancer that I highlighted here, so proliferation, invasion, resisting cell death, and replicative immortality in cell culture models, and I'm just summarising it here because I appreciate that it's quite complex. So in our at-risk model, brake off, accelerator off, what we see is increased proliferation. That's what I've just shown you. It has no effect on the viability of the cells, so it doesn't impact on this pathway. We have increased sphere forming efficiency, so this relates to replicative immortality. Basically, it means every cell in the culture is capable of forming a cancer cell. The the sphere's size increases, we've got more neurospheres and we've got more invasion in this model. Again, contrasting this with the high grade cancer, which is the accelerator off and the brake off and the accelerator on, it's the opposite. So again, expressing the prion protein, you get reduced proliferation, you get a trend to reduce viability, you get reduced neurosphere size and a trend to reduced invasion. So in this context, in the high-grade cancer model, PrPC expression knocking out PrPC expression is protective, and in the low-grade model, knocking out PrP expression increases the risk of cancer. But this is all in a cell culture dish and doesn't really represent what's going on in an actual brain of an individual. And so using the same mouse model, what we can do is turn on those cancer genes. So essentially the way this works is we've got all those different mice that I spoke to you before. So I've got the brake off, accelerator off, brake on, accelerator on, all the different combinations. And basically what we can do with these mice is we can turn on those genes. Again, we do this by treating them with this drug called tamoxifen. 3 minutes. Okay, that's all right. And basically what we've done is take those mice out to either 6 weeks post-cancer induction, 22 weeks post-cancer induction, or 42 weeks post-cancer induction. And these are the tumours that these mice grow. So this is the brake on, accelerator off. So this is the normal. The tumours grow in the ventricles of the mice, and there's no tumours in somebody who's not at risk of cancer. This is the brake off, accelerator off. So these guys are at the low risk of cancer, and you can see these small tumours growing in the ventricles of these mice. And then this is the brake off, accelerator on, very aggressive tumour that you can see have filled the ventricles of the brains. So what we see when we compare the different genotypes of the mice is that in the PrP, this is in the low-grade cancer model, when you have PRP knockout mice, just like we showed in the cell culture dish, the tumours are larger than mice that express wild-type PRP. So where we're going with this work now is, so the high-grade model, we've only just recently induced those tumours, so we haven't had a chance to analyse it in detail. But what we have been, I'll show you one other thing. When we look at the morphology of the tumours, not only are they larger, but they're more cellular. So there's clearly something going on at the molecular level in these cells, in these tumours as well. But my last slide is just to make the point that what we are doing is this technology called Visium spatial gene expression. So essentially, Shana Patel, who I mentioned before, who is the PhD student on this, also received a fellowship from 10x Genomics, which is the company that develops this product. And the benefit here was that the dollars that you donated, we were able to double the value of them through this fellowship, because the fellowship gave us reduced pricing on this very expensive technology. And basically what we've done is, or we'll be able to do, is look at individual genes expressed in different regions of the mouse brain and the mouse tumours to understand molecularly what is driving this difference in tumour progression, in whether or not we have a knockout or a wild-type expressing mouse brain. So far we've prepared the samples, we've done the workflow, we've generated the libraries, we just need to do the sequencing and the analysis. And so I'll just finish up by acknowledging the support that we've got through the CJD Foundation here in the US, again through the strides of CJD research and the CJD Foundation research, but also to acknowledge the CJD support group network in Australia, who have provided a top-up scholarship to Shana and also gave us some seed funding before we received the CJD Foundation grant to support this work.
Thank you very much. So we're going to move on, and if you've got questions, make a note of them. Next person is Ignazio Cali from Case Western, Cleveland. And again, I'm just going to put a bit of context around it, I hope. Neurodegenerative diseases are familiar to neurologists. They're a group of diseases that include Alzheimer's disease, Parkinson's disease, and Prion disease, and others. And they're basically defined because certain neurons, certain nerve cells degenerate and die, particularly in the brain, and this is associated with abnormal folding of very specific normal proteins. In the case of prion disease, prion proteins. And these proteins, when they're abnormally folded, are deposited in the brain tissue. And they are distinct diseases associated with distinct proteins. But pathologists, I think, have become increasingly familiar with finding more than one kind of protein pathology in an individual person's brain. And this raises questions as to how these different proteins might interact in disease. And indeed, I think it's important to emphasize that proteins are very mobile things. They're not these kind of static pictures that you see on the screen. They are created, they live a short time, they're destroyed, recreated, they move around, and they interact with all sorts of things. They're not just individuals. So this presentation is about exploring possible interactions between prion protein and other proteins in different diseases.
Good morning, everyone. So the title of my presentation today is the detection of abnormal prion protein in patients with non-prion disease dementia. So major questions of this study are to determine whether the abnormal form of the prion protein accumulates in the brain of patients with Alzheimer's disease and frontotemporal dementia. So we also wanted to determine whether PRP codon 129 and the ApoE genotypes, as well as the age and the rate of disease progression, affect the formation of small PRP aggregates, which we call PRP seeds, whether these PRP seeds are present in different brain compartments or only in specific brain regions, and also whether PRP seed activity, which we measured by RT-QuIC in these patients, resemble that of the human sporadic prion diseases. So again, for this study, we used brain homogenate from deceased patients, and the real-time QuIC-induced conversion to determine the presence of misfolded PRP. As you know, the in prion disease, the conversion of the cellular or normal prion protein or PrPC into PrP-scrapy is a spontaneous conversion and occur. I mean, this is a very rare event. However, we can mimic this in vitro. As you can see here, what we do is just to mix the normal PrP, which has been expressed in bacteria, which we call the substrate, and then we use a diluted brain homogenate to determine whether there is or not PrP, I mean misfolded PrP, and the thioflavin T, which will eventually binds to the growing amyloid. So the microplate is incubated, and again, if there is a seed, PrP seed in our brain homogenate, then so basically we have a seeded conversion, and we will get this red profile, which there is an exponential growing of the seeding activity, which is preceded by a lag phase, which can be long or short, and depending on the amount of the prion protein or the strain of the PrP. Here are methods. We have studied 100 cases of Alzheimer's disease, and we tested 505 samples of these cases. And you can see the age is 80 plus minus 10. I'm talking about the mean plus standard deviation. So 9 cases of frontotemporal dementia with age of 71. So these are what we call non-demanded, the negative controls, and we divide in 2 subsets. So young patients, we have 20 cases with 39 years old, and all patients 45 with which are age matched to the Alzheimer's disease and FTD cases. And of course, we didn't know anything about whether there would have been seeding activity, so the formation of PrP seeds in these patients. So these are 3 other cases. One is primary age-related tauopathy, metabolic disease, and multiple system atrophy, for which we know there were inconsistent PrP seeding activity. And these are the positive controls. So what we used, we are here sporadic CJD. So we use like 2 subtypes, SCJD, VV1, SCJD, MEM2, which are known to have like a non-efficient seeding activity, and sporadic, as well as sporadic fatal insomnia and VPSPR, which is another sporadic human prion disease. How many brain regions we have tested? So in 28% of the cases that we have studied, we only had 1 brain region, and typically it was the frontal cortex. In the great majority, 62%, we tested 2 brain regions, the frontal cortex and the cerebellum. So in 5%, we had 3 brain regions, which is the frontal cortex, or the parietal cortex, a subcortical region, which would have been the thalamus, the putamen, and the cerebellum, and then 5% of the cases in which we have between 5 and 12 brain regions. We have a large sets of samples. So, and here, this slide is just to tell you about basically what we call positive and what we call negative on a single experiment. So we call, of course, negative when an experiment which is done in quadruplicate. As you can see here, we load the same sample in 4 wells. And so basically, if there is no replication, then of course, this is negative. If there is only 1 well, if the seeding of the PrP is observed in 1 well, we will call this negative. And we will call this positive if we would have at least 50%, so 2 out of 4 wells positive, or 3, and of course 4 wells. Here are a few examples of what we did. So basically, you can see here we have case 1. We took the frontal cortex. All the 4 wells were positive on the first experiment. We did the second experiment was negative, and we called it negative. We wouldn't continue doing other experiments. This is the cerebellum, the same case. These are just examples. So this is negative. 1 out of 4 wells one well was positive, but and here 1 well was positive. But if you remember about the previous slides, anyway, this case would have been called negative, and negative is this one, this case 2. However, in the cerebellum case 2, you can see that on the third experiment we have 2 wells positive, and then this case would have been called positive in the cerebellum. And here is the degree of the positive outcome. So we could have a positive case. We would have done, let's say, 4 experiments, and the positivity was only on the first experiment, and so the degree of positive outcome is low, so low seeding nativity. The same for this other case, which has 3. Here is positive on the second case. Here is moderate. We run 5 experiments. It was positive on the first experiment, positive on the third experiment. Moderate, here we have 2 positive experiments, the first and the fourth. And high, so 3 out of 5 experiments are positive, 4 out of 5 experiments are positive. And there are also cases in which we did 5 experiments and all of them were positive. We have very high or robust PrPC nativity. So what we have done, we looked at the genetic, so that's genotype codon 129 and ApoE, which is a risk factor for Alzheimer's disease. We are looking at the mutation PP, presenilin-1, presenilin-2, which leads to familial or genetic Alzheimer's disease. We are looking at the phenotype and the histopathology. We're looking for the presence of spongiform degeneration. We have done immunohistochemistry for PRP, amyloid beta, hyperphosphorylated tau, and other proteins like alpha-synuclein TDP-43, histoblot, which is basically an immunohistochemistry on frozen tissue. And of course, we have checked the presence of the PK-resistant, the prion protein on Western blotting. And finally, we have used a transgenic mouse model of comorbidity. So this mouse model basically expressed the human wild-type prion protein and the human mutated APP, which cause Alzheimer's disease. And so the difference between these 2 TG mouse line is the genotype codon 129, which is a methionine homozygous in this mouse line, and the VV or valin homozygous in the other one. And then, so basically we took old and young and old mice, we took the brain, we harvested the brain, and we dissected it in 2 major compartments. The cerebral cortex, it contains the hippocampus and the rest of the brain, so the central brain, the brainstem, and the cerebellum. And then we ran the RT-QuIC on these mouse brains. And here you can see, now talking about the results, the prevalence of cases with positive PRP RT-QuIC, and we are comparing with demanded and with ND not demanded cases. You can see these are age-matched and these are young patients. And so basically, the prevalence is the same between demanded and non-demanded. Demanded are Alzheimer's disease and frontotemporal dementia. These are age-matched cases without dementia. And it's striking that basically cases with that are not demanded, that are young, have no seeding activity at all. So, however, let's look at the outcome of the positive results. Is it high, moderate, and low? So if we look at the high robust PRP seeding activity, you can see that only the demanded cases with 37% and none of the cases that are not demanded have PRP seeding activity. However, so there is no difference when we go down to cases with moderate or low seeding activity. There is no difference. The only difference is here in the high degree of positive outcome. So the next slides is about, we checked about what does the age has an effect on the PRP seeding activity. And you can see here, and we are looking at different diseases. This is Alzheimer's disease, cases without PRP seeding activity and cases with a positive PRP seeding activity. And you can see there is a little, a younger age is not statistically significant of the AD cases with the positive PRP. And these are the FTD, is the same trend. These are not demanded cases. However, if we would combine this and to increase the power of the experiment, you can see that the cases with positive RT-QuIC are statistically younger than the cases with the negative RT-QuIC. And then the next slide is about the disease duration or the rate of disease progression. And you can see that basically cases of, and of course we look at the cases of Alzheimer's disease, which we had 100 cases, and we had cases with short disease duration, which means high disease progression, and cases with very long disease duration, the classic Alzheimer's disease. And you can see that there is a subset of cases with the cases with positive RT-QuIC are those that have a faster disease progression. Let's look at we look at the lag phase as a kinetics of PrPC information between AD, FTD, and non-demanded cases. We found some differences here. So the cases of Alzheimer's disease have a slower, a longer lag phase compared to the other two groups. And we compared, anyway, this AD and FTD and the non-demanded cases with the cases of sporadic prion disease, which have anyway, even if they are recognized like being like having an inefficient seeding, they seed faster and they have a shorter lag phases compared to the other groups. Here what we looked is the difference between cortical and subcortical. So cortical is typically, we have a shorter lag phase compared to the subcortical. We looked also the cerebellum. The cerebellum, basically, we, apart from two cases, we never saw any seeding activity. However, I think that this lack of seeding activity in the cerebellum is due to the low amount of total of the PrPC as we measure this on Western blotting. And this is, I think, is a very important slide because it shows that cases that are homozygous Codon 129 have the shortest lag phase and increase on MV and the VV. So we found no difference on ApoE genotype. These are AD, all cases AD with a positive RT-QuIC with or without E4. You see there is no difference. The same for the FTD. And the last slides, so basically mice with MM have a PrPC inactivity, mice with VV didn't have a seeding activity. However, if for a second we forgot what I said before and we consider positive also positive the mice that have also one single well, seeding activity in a single well, we would have 3 mice with seeding activity, and again, like we have seen in a human, they have a longer, a greater lag phase compared to the MM, and also mice with MM genotype, so tend to misfold in the cerebral cortex rather than subcortical regions. Conclusions are that a robust PrPC activity is found in demanded case, but not in age-matched and not demanded controls. These results point to the deleterious effect of a primary proteinopathy leading to disease comorbidity. PrP seeding is age-dependent as young and non-demanded controls are free of PrP seeds. AD cases with rapid disease progression, as we've seen before, that have disease duration less than 3 years, and positive PrP seeding were 7 years younger than classic AD. So therefore, age and disease progression at the different levels, I believe, may impact PrP seed formation. So patients with PrP 129 MM genotype may be at higher risk of developing for PrP seed formation. So the short lag phase in the cortex and the virtually lack of PrPC in the cerebellum may be due to the differences in the amount of PrPC that is available for PrPC to PrPC conversion in this brain compartment. And finally, in patients with AD and FTD, the presence of a misfolded PrP may represent a very early stage of prion disease comorbidity. The future direction is to investigate whether these samples with positive PrPC nativity is infectious when they are inoculated to transgenic mice that are susceptible to the prion protein, abnormal prion protein. I would like to thank the CJD Foundation, the Jeffrey Mary Smith Family Foundation, my former lab in Case West, San Antonio, Cornacchia, Liam Weisinger, and Sansu, collaborators Case, Xiongwei Zhu, Zerui Wang, Witold Surovic, and the last slides, I just recently moved to Italy. I'll be in Enna, in Sicily, but I will be still collaborating with the surveillance center. Thank you.
Thank you. So our last presentation is Sarah Vascellari from the University of Cagliari in Italy. And again, I'm just going to give a bit of brief context beforehand. As you'll even be tired of hearing perhaps now, the core molecular event in prion disease is the formation of abnormally folded prion protein. So detecting this protein is really the gold standard of diagnosing these illnesses, and it certainly forms the core of neuropathologists looking at the brain and finding that abnormal protein, saying that that's what it is. But all detection methods have a sensitivity. They require a certain level of prion protein. And of course, the brain is not terribly accessible for clinical diagnosis. And if indeed prion protein was found in other tissues in the body, abnormal prion protein, as indeed it is, it tends to be there at very low levels. But people who are much cleverer than me have developed amplification methods which allow low levels of prion protein to be amplified to the point where they can be detected. So clearly, if this could work, as it has done in spinal fluid, for the RT quick test in spinal fluid, if it could work in more accessible tissues like blood or urine, that would be very helpful.
Good morning. Thank you for the presentation. It's an honor to be here. My presentation today is aimed to give you an update on our project, which focuses on development of a blood and urine prion diagnostic test for human prion disease using RT-quick assay. The ability to diagnose CJD in live patient has improved over the years. Over the years, from the detection of specific changes in brain magnetic resonance imaging and electroencephalograph, to include the detection of biomarker in cerebrospinal fluid, such as 43 protein and the prions using the RT-quick assay. The RT-quick assay is being developed by Cogis Byron's group for the amplification and detection of prions in biological sample. The test involves the use of recombinant prion protein produced in bacterial cells to amplify a very small amount of prions in biological specimens. The reaction is done in a small vessel in a plate with 96 wells, and the reaction go through cycle agitation and the rest in an instrument called plate reader. The reaction are performed in presence of tyrflavin T, which is a fluorescent dye, and this dye binds prions that amplify in the wall and organize themselves in a structure called fibrils. Once bound THT emit fluorescence, and we can detect the amplified prion from positive sample. The RT-quick assay has been adapted to different biological samples. For example, olfactory mucosa obtained with nasal brushing, or CSF that is collected through a procedure that called lumbar puncture. Routinely, CSF is collected in case of rapidly progressive dementia to rule out, for example, viral infection. But there is a great interest to use alternative sample that can be collected with less invasive procedures, such as blood and urine. Animal studies confirm the presence of the prion infectivity in blood and blood fraction of sporadic CJD patients, and we know that the higher level of infectivity are associated in descending order with white cells, whole blood, platelets, red cells, and plasma. In our experience, main issue testing the blood sample are the low level of prions and the inhibitory effect that some component of blood can have on the RT-quick assay. To overcome the issue, Oru et al. Developed a quick assay, or enhanced quick, using an anti-prion antibody called 15B3 associated with magnetic beads to recover and concentrate and isolate the prions from plasma, and RT-quick to amplify the capture prions in the sample. We use this approach. Sorry. So we use this approach to test plasma from sporadic CJD patient with different protein genetic profiles, and our results show the detection of prions in 5 out of 7 sporadic CJD sample. Positive sample. Okay. Positive sample were subtype MM1 and V1 and VV1, while the negative were MV2 and MM1 subtypes. Unfortunately, this antibody is not commercially available anymore, and we have been working on developing alternative strategy to detect prions in urine and blood sample. Several methods for prion capture and detection is being described. Given the ability of some metals, the ability of prions to bind some metals, some authors used iron oxide beads and steel beads to concentrate azorin prions from sample. And Lawson group used nanoparticle to overcome the effect of inhibitors of the RT-quick in complex tissue sample. So we evaluate whether the iron oxide, don't know why, okay. Steel beads and iron oxide beads, iron oxide bead alone in combination with steel beads, could be used to detect prions from a blood sample. In particular, we focused on whole blood that contain white cell, red cells, and platelets and plasma. and a Buffy-coat fraction that contain white cells and plasma. We mix this sample with iron oxybeads and steel beads, and we recover the prion using a magnetic rack, and after we put this in the reaction in the presence of nanoparticle. In our result, testing whole blood and Buffy coat combined with no amount of prions, sporadic CJD prions brain tissue, show the combination of iron oxide beads with steel beads allow it to recover prions from the sample at rate extreme dilution, 1 billion-fold dilution for Buffy coat and 10 million-fold dilution for whole blood. Then we evaluate if whether iron oxide beads could be used to isolate the concentrated prions from the urine. And first, we combine the urine with a known amount of prions from brain tissue, and we combined with iron oxide beads. We recover the beads with a magnetic rack. We put the beads in one well or split them in 4 wells. And our results show that iron oxide beads allow to detect prions in urine, even when the amount of prions added was extremely low, until 10^11 brain tissue dilution. Then, when the reaction, a second round, the reaction was performed, transferring a portion of reaction mix for the first reaction in a new plate with new reaction mix, we observed a faster and more robust amplification. You can see here at this dilution minus 2^11 brain tissue dilution. And also we try to increase the concentration of iron oxide beads in the sample to capture the prions. And after that we recover the iron oxide beads with the magnetic rack, we transfer all beads in one well and in the presence of nanoparticle. But we observe the inhibition of the reaction after the first round of the reaction. Performing a second round the reaction, transferring a portion of the of the reaction mix from the first reaction into a new plate with new reaction mix, we overcome the inhibition, and we observe faster. It doesn't work. Faster pre-amplification and increase of reaction sensitivity until 10^13 brain tissue dilution. Overall, our data show a promising approach based on metal beads to capture prions from blood and urine sample combined with brain tissue dilution from sporadic CJD patients. This strategy may improve sporadic CJD detection in blood and urine sample. I would like to thank all study participants, the co-investigator of this project, Cristina Ru, Byron Coy, Aldo Manzin, and all collaborators that who provide the sample, Franco Cardone, Gianluigi Zanusso, Pierluigi Garbetti, Stefano Eich, and Larisa Cevernakova. And also Andrea Astion for providing the recombinant prion protein, and Bradley Groverman to the graphical assistance. A particular thanks to the families that have contributed to fund this project. Thank you, and also thank you, Debbie Jobs, and also whole member of the CJD Foundation, and also thank you for your attention.